Let me know if it has any more problems. Thanks, Charles. Can you see it? Okay. All right. So with qPCR, we're going to measure this amount. And but you can see here is that the target input here, the one in red is 10 to the 8th mRNA molecules. In this one, in orange, is 10 to the 7th, then 10 to the 6th, 10 to the 5th, 10 to the 4th, 10 to the 3rd, and then just 100, and then 10 right here, and then no molecules at all. And you can see with the cycle number here, and then we're going to call this right here. This is where we're going to do our reads. But if you look at the differences here, we're really, here we're at, we have liftoff at cycle 24. Here we have liftoff at cycle 27. So in other words, we don't see anything, but then by the time we get to cycle 24, we begin to see a band showing up. And here, with this amount, we begin to see a band showing up around cycle 27. So it's only 3 cycles difference. And you can see if we looked at this, if we stopped right here at this particular cycle, at cycle 30, we would see a difference between these two, but we wouldn't see much of a difference between these ones, would we? And similarly, if we went down here, we would see a cycle between 1,000 and 100, but we wouldn't see much difference between 10,000, 100,000, and 1,000,000 at all. So how do you know exactly, in the old days when they were doing PCR and they tried to do it quantitatively, they would have to stop it at every cycle and then run it on a gel and then figure out where it's going to happen, because depending on which cycle you stop it at determines how much you're going to see, and if you're going to see a difference between one amount and another amount. And mind you, these amounts are orders of magnitude different, right? Each one is tenfold different. So using qPCR to estimate the concentration of mRNA was a very dicey proposition. So what they did instead is they invented a machine that would have, in the PCR, you have the place where you put your tubes. Underneath each place which had the tube, they put a detector which would detect fluorescence. So you can keep track of the fluorescence of every tube by automatically. And in these qPCR machines, every tube is tracked for fluorescence. And you wind up coming out with a diagram like this. Okay? So the only thing you have to do is you make a standard curve, which this is, and then you have to decide, okay, where are we going to measure the cycle number? So you can measure the cycle number right here at 500, or you can measure the cycle number down here at 100, whatever. You choose where you're going to measure that cycle number, and then you drop the arrow down, and you say, okay, that's the cycle number. And that's how you make a curve between the target input, the number of molecules, and the cycle number. And then you wind up with a nice, pretty graph. Let's see if we can find a pretty graph here. Which is your standard curve here. So here you take this information on the left, which we just saw, and then you make a standard curve, the quantity of it versus the cycle number, the CT. Okay? And that's basically it. There's your standard curve. And then whatever your sample is, you just go ahead and run it around this, you measure the CT, the cycle number, and then you go to your standard curve and you can figure out how many molecules of mRNA you have. And that's basically how it works. Any questions then about qPCR? How that works? Also, if you use Cyber Green, Cyber Green is the cheapest way to do it. There are more expensive ways, but let's look at that real quick. There is a, oh, here it is, back here. Oh. If you use Cyber Green, then you can go ahead and denature it as you would for PCR. And then you add your primers, and you add the tack. And then, this is annealing, and then you bring it up in the temperature, and we do the normal PCR. And then you can see these little green, cyber green dyes here. The cyber green dyes will bind only to the double-stranded DNA. That's what they do. They bind to double-stranded DNA. So the longer you make your strand, the more cyber green is going to bind. And so it's quantitative. The stronger your fluorescence with cyber green, the more DNA you have. So fluorescence equals DNA concentration, and that's what you're looking for. Okay, thank you very much for that. So let's move on then to flow cytometry. And I found a really good site for this at University of California, San Francisco. And they have, just for their training, they have something called iRocket. And so we'll go and study flow cytometry from iRocket. And, oops, I don't want to do that. I want to go over here. Visit page. Okay, so you could go to the UCSF website, and they give you a very nice explanation on how flow cytometry works. The basic idea here is that you want to take individual cells, and you want to do a measurement on every last cell. You want to do a fluorescence measurement on every last cell. So what that means is, let's say that you're taking Lake Waco water, which is a wonderful pea green, and it has lots of different microorganisms. And let's say there is a surface antigen on a particular organism. I don't know, let's pick Salmonella, or maybe Giardia, your favorite pathogenic microorganism. You take an antibody against that surface antigen, and then you go ahead and take your Lake Waco water, you add the antibody, and the Salmonella is all fluorescing because it has that antibody on it, a fluorescent antibody. Then you take it and you run it through this flow cytometer, and it individually takes your little bacteria, the individual bacterial cells, and it puts them in individual droplets. So one bacterium per droplet. And then it goes down here, and then you have a fluorescence detector which will read whether you have fluorescence or not. And then, of course, you have laser excitation. And that measures the amount of fluorescence in every drop. And then you can figure out what proportion of the cells are fluorescing. And if you say, well, only 1 in 1,000 of my cells are fluorescing, you say, well, there aren't many Salmonella in Lake Waco water. But if it's 1 in every 4, then there are a lot of Salmonella in the Lake Waco water. Another thing you can do is you can have this thing called scatter detection. And scatter detection can measure lots of different things. So, for example, with scatter detection, side scatter, SSC, you could measure the volume of the cells, whether they're bacteria, which would be Salmonella, or bigger, like Giardia, which is a protist. You can measure DNA and RNA, all sorts of different things. So that's with side scatter. And if you are using a fluorescent antibody, you can measure surface antigens, and many more, as they promise. So a lot of things you can do with this. Now, another thing you can do is called fluorescence activated cell sorting, or FACS. And with FACS, what you do is you go ahead and take your sample and you do the same thing you did before, but this light source, which will tell you the fluorescence, that fluorescence detector is connected to a computer which will connect to these deflector plates, positive, and negative deflector plates. And those deflector plates will throw some cells, which might be fluorescent, into one tube and other cells into another tube. So let's say we had a positive and negative deflector plates here. And let's say you had some cells which are labeled red and some which were labeled green. This goes through the light source and it fluoresces green. And so the computer knows it takes 4 milliseconds to go from this point down to this point. So in 4 milliseconds it says fire, and then this thing has a charge 4 milliseconds later, and that exact drop gets flicked into this tube. This red cell is going through and it yells out fire, and by the time it reaches here, this one has been activated, and that exact drop with the red fluorescent bacterium gets flicked into this tube, and then all the rest go into this waste tube. So it is a fluorescence-activated cell sorter. So let's say you were doing something like, I wanted to grind up spleen cells, or lymphocytes, some of the lymphocytes, and I wanted to find all the CD4 positive cells. Well, what would you do if you wanted to, where does CD4 live on the cell? Is it in the nucleus? The mitochondria? Where is it? It's a surface, yeah. So what could we do to identify CD4 positive cells?
Bind an antibody.
Right, we'll use a fluorescent antibody. So you can go ahead and get a complex tissue mixture, add the fluorescently labeled antibody against CD4, then run it through here, and then bang, you can collect all your CD4 positive cells from a very complex tissue. It's incredible. When you stop to think about it, it's microdissection. So that's a very nice thing. Now, when we go ahead and look at the output, here's the output. You don't have to use a cell sorter, but you could just measure the output. And if we have two different ones we're looking at, two different fluorescents, one is red and one is green. We'll use a antibody against CCR7 on a surface antigen, and we'll use, let's say, a red fluorescent antibody for that. For this one, for CD45 RA, that surface antigen, we will use an antibody which has a green fluorescence. So we run it through this, and it keeps track of red fluorescence and green fluorescence for every cell. And it makes a mark. Okay, that was red fluorescent. Oh, that one was green fluorescent. Oh, that one didn't have much fluorescence. And on top of that, it says, wow, that one was really green, or that one was just a little bit green. That one was very green, but a little bit red. That one was very red, but a little bit green, right? So it keeps track of all those things. And of course, you have two parameters. How red is it? How green is it? Well, how red is it is kept on the x-axis. How green is it is displayed on the y-axis. And you put a dot for every cell you observe. And in the end, you wind up with this scatter plot. And then you can divide up the scatter plot depending on your own thresholds. And here we seem to have 4 populations, so it actually looks like one big blob. But normally you'll actually have 4 blobs or 2 blobs or whatever you're looking at. And let's say this were really 4 blobs. We'll color this blob green, that population green. We'll color that population red. We'll color that population some other color. And then this population will purple, and then we'll color this population blue. And then